If we can kill off senescent cells in our body, we will grow younger. But, we don't want to kill the rest of our cells, obviously.
I'm here to kill your cells.
I need those to live. This paper by Poblocka et al., they describe a way to accomplish just that. In cells. In a dish. It's not in people yet, but it's an important first step, and I will explain why. I'm Dr. Peter Allen. I'm a bioanalytical chemist and host of this science YouTube channel. In this video, I want to go through a peer-reviewed paper I found really interesting. It's about a new technique to kill senescent cells, and it's a step toward managing, slowing, or even stopping the aging process. As we get older, we accumulate senescent cells. Senescent is a word that literally means old, but in cell biology, it means a very specific thing. The cells are not dividing, they're not dying, and they're not doing their job. Senescent cells are part of the reason why we have health problems as we get old. They disrupt the immune system, they hog resources, they're generally a bad thing to have hanging around. A few years ago, a group of scientists engineered a strain of mice with killable senescent cells. They let some of those mice grow up normally, and they didn't have any particular problems. They were perfectly normal mice. They died like regular mice after about 3 years. They took a group of those same mice and used their modified genes to kill the senescent cells, and the mice lived longer and looked and behaved healthier. It was as if they aged in reverse for a while. In human tissue, you can make senescent cells turn blue, and they have this enzyme that most other cells don't have. It's a beta-galactosidase, if you're interested. It might seem like you could make a drug that's activated by beta-galactosidase, and that might be able to kill senescent cells, but that enzyme is inside the cells, and the enzyme's not totally unique anyway. That poses two problems. The drug would have to get into all the cells in order to get the enzyme to chew it up, and that would have to kill just the senescent cells. And some cells have a form of beta-galactosidase as well, and those would be in trouble too. So what else could we do to target a drug to kill just senescent cells? It'd be nice if we could find a specific marker on the outside of senescent cells that we could latch onto in order to target the cells with some drug. And that's exactly what Poblocka et al. Did. They found such a marker called B2M. Now, if we could kill cells that express B2M, that have it on the outside of their surface, we could maybe de-age humans the same way we de-aged those genetically engineered mice. Because B2M is on the outer surface of the cells, that means that there's a handle in the blood to latch onto, to grab these. To make a little analogy, it's kind of like the Death Star's thermal exhaust port. It's a vulnerability right on the surface, right out in the open. Things flowing through the blood can get to it. It's not hidden away inside the relatively protected interior of the cell. Now we need an X-wing, and that's an antibody. Antibodies are shaped like a Y-wing, actually, and these antibodies can carry a weapon. They can attach a drug to the outside of the antibody that can then target that specific vulnerability. That's the proton torpedo in this scenario. The idea is if you can find a senescent cell's vulnerability, you can use the antibody to target it, get the drug into the senescent cells and just the senescent cells, and then kill them. Even though it's a very powerful weapon, it's not going to kill anything else. This is called an antibody drug conjugate, and that's what's discussed in Poblocka et al. So this is the paper. It was published in 2021 in Nature Scientific Reports. I like this journal because it's an open access journal, meaning that you can get a copy of it for free without having to be at an institution, a university, or research institution. This publication came out of a collaboration between the University of Leicester and the University of Barcelona. Lead author is Marca Poblocka. I won't go through the whole thing, but the key is to look at this figure. So the key finding is that P16 is an age-related senescence marker, and we get more of it. It gets darker with increasing age. And B2M, their new marker, follows the same pattern. This is all in skin cells, and this down here is in brain cells. And if the images are a little unclear, you can see that the same thing happens in a quantitative metric. So if you look at how much dark color there is using image analysis, you can quantify that we see more as skin and brain cells get older. So now that we know that this molecule, B2M, is a senescence marker, we can show that as you add an antibody drug conjugate, that's ADC, cells become less viable. That is to say, these numbers go down as you increase the ADC concentration. Now, I'm not going to go through every result in the paper, but that's the key, is that one, this marker is on the surface, and two, it is targetable with this antibody drug conjugate. Now, there have been some small molecules that have been used to target senescent cells, but they haven't proven to be, well, they haven't passed FDA approval yet. So it may be that this is the beginning of an alternative approach. And this is just the beginning of synthetic antibody drug technology for killing senescent cells. This particular paper doesn't cover animal studies, much less human studies. We're a long way from actual medicine, but there have been clinical trials on small molecule drugs trying to do the same thing. So if the next generation of small molecule senescent cell killers don't work, well, this is an interesting alternative approach. This is just another example of the kind of technology that might produce the breakthrough I'm hoping for in anti-aging. So please keep coming back to maybe even hit that subscribe button, and I will keep making these videos on anti-aging technology. Would you like to see a video about some supplements that might have a small effect and you could actually try them right now? Check out my video on nicotinamide adenine dinucleotide supplements in the link below and up here. And as always, you can visit peterallenlab.com for shareable graphics and references.